Hope you enjoyed Dr. Porterfield and I having a bit of a conversation here. Really want to reiterate what a great year we're having for our grand rounds. Thank you again to the support of our chair, Dr. Mosley, for all of our educational excellence to move forward. For our Roy Benke Department of Internal Medicine grand rounds for this Thursday, we have Dr. James Zachary Porterfield. He goes by Zach. Dr. Porterfield spent what must have felt like an eternity at the University of Oklahoma, where he graduated with his Bachelor of Science in Physics, Zoology, and Biomedical Sciences. So cum laude in physics. So, and from there continued his studies in Oklahoma, becoming an MD PhD with his full MD graduation as an MD PhD in 2012. Then went to something that we're very much talking about with our MD PhD program here at USF, and had the pleasure of meeting the three MD PhD students of our College of Medicine on Monday. But we were talking to them about the physician scientist pathway that's out there. And here is Dr. Porterfield at Grand Rounds as a very successful physician scientist pathway graduate. Dr. Porterfield did this at Yale, his residency and his ID fellowship there. Then ended up going to the University of Kentucky and spent about 4 years there before we, and by we I mean Dr. Kami Kim, grabbed him and recruited him to come to USF in the fall of 2023. The accomplishments of Dr. Porterfield would take me, I think, about 40 minutes to go through, so I'll just summarize very shortly with some highlights. NIH Fogarty Global Health Fellow, which included a stint with the African Health Institute for Research. And he did that for about 3 years. He's had numerous chair positions, chair of the Global Virus Network for Retroviruses Action Committee. That's a mouthful, and I can imagine how much work went into that, just having to try to state that committee name. And he also was the chair of the subgroup on otitis media for the Lancet Commission on Hearing Loss, that's a fascinating subgroup that you had to work on there, Dr. Porterfield. But I also want to highlight, he was one of the forefront authorities in the National COVID Collaborative Therapeutics Working Group, and gave one of the first grand rounds possibly in the country on COVID-19 in the spring of 2020. And his publications and presentations, like I said, I'd be here probably already 35, 40 minutes doing. So with that, I'm going to turn it over to Dr. Porterfield. And I really, like I said, it's been a pleasure to get to know Zach in preparation for this and to review all his accomplishments. I'm really looking forward to his talk on the impact of HIV reservoirs on adaptive immunity. So with that, Zach, welcome to our grand rounds, and we look forward to this talk. Go for it.
Thank you, Dr. Lazama. I really appreciate the opportunity to present in this forum. I want to thank Ms. Jennifer Newcomb for all of her assistance in preparing and helping me get ready for this talk. So again, we're going to focus on HIV reservoirs, on adaptive immunity, which is the focus of the work I do in my lab and in my translational life.
And everyone can see this. I want to say Zach is coming off of a trip to New Zealand, and for those who didn't hear our conversation earlier, a flight that got diverted because of a stroke patient. So he's had a journey to make it here. So thank you again, Zach. And absolutely, just want to share that background with you. So take it away.
Thank you very much. So one of the nuances of the work that I do is I am on a lot of airplanes. So I have a large global health focus in my career. So I'm a physician scientist. My PhD training is in virology. I chiefly focus on viral infections in tissue. A majority of that is HIV, but we also work on human papillomavirus and its intersection with oropharyngeal cancer, and certainly have done some COVID work in my time as an independent investigator. As an immunologist, I'm really interested in the effect of HIV and other viral infections on the immune response in humans, and specifically looking at that response in tissue, which is a unique angle we're going to talk about. Clinically, I'm board certified in internal medicine, practice here as an infectious disease physician. And we also, in my lab, conduct quite a bit of clinical research focused on HIV, TB, and some non-communicable diseases, and then have an implementation focus. And a lot of this really revolves around a long-term focus I've had in global health, both in international exchange and capacity building. I've worked and lived in South Africa and Sub-Saharan Africa for about 25 years off and on, and have some very active and healthy partnerships and collaborations in Sub-Saharan Africa. Things that started when I was a resident and fellow at Yale, and have transitioned with me at the University of Kentucky and now at the University of South Florida. So I think we've already brought, gosh, maybe 24 trainees and students back and forth from the University of South Florida. But that's a major focus. And I think it really brings together all of these various different aspects of the work that I get to do. So I really believe in the mission of physician scientists. And I speak enough clinical medicine and enough research to, I think, bridge some of the gaps. And that's a position that I feel very fortunate to get to do. So my lab is currently here at the University of South Florida in Research Park. It's a beautiful new research building. If anybody is interested, please feel free to come by for a tour, and certainly we can put you to work if anyone is really interested in doing some pipetting. We also have a lab in Durban, South Africa, which I'll talk more about here in the next few slides, and that I've been actively involved in for the last 10 years, working in the research facilities in Durban and in the Western Cape. So I just want to take a little bit of time. So we'll do some preamble slides to just think about HIV and the global HIV epidemic to set the stage for the approach that we take in our lab. So there are, currently 38.4 million people living with HIV, and there are new acquisitions of HIV. So although the rollout of antiretrovirals has been incredibly successful in turning the tide and controlling the epidemic, there are still people who are acquiring HIV, both through sexual contact, IV drug use, and also vertical transmission to children. And there are a large number of deaths from individuals living with HIV. Still every year. So while this is not the same disease it was in the late 80s and early 90s, it still is a global epidemic, and a large burden on the populations that are living with HIV. So even though we now have very highly active antiretroviral therapy, continuing to study this epidemic and thinking through ways to take better care of the individuals who are living with HIV is important. It's also really notable to say that we now have an aging population living with HIV. So for the very first time, we have people who are, large portions of the population living with HIV who are in their 50s, 60s, 70s, and even 80s. And so understanding the intersection between HIV, whether it's controlled on antiretrovirals or not, and long-term disease and immune senescence, I think is a really interesting challenge as a clinician, as a researcher. I want to start briefly by talking a little bit about the Africa Health Research Institute, which is where I spend a lot of my time in South Africa, and I travel 3 or 4 times a year, usually for very short durations, but to continue the research projects that we have in collaboration with ARI. And this is now often happening with infectious disease fellows, public health students. We have ENT and audiology students who come with us, but we are bringing individuals back and forth, and in fact currently have some students from Malawi who are visiting the University of South Florida. But ARI is comprised of two different facilities. One is a basic science research facility, which is seen on the left-hand side, and this is a world-class HIV and TB Research Institute that was initially endowed as a collaboration between the government of South Africa, the Doris Duke Foundation, Howard Hughes, Wellcome Trust, and is truly a remarkable place to do HIV research. And the second half of this is a clinical research facility in Somkhele, which is about an hour and a half north of Durban, which is a remarkable clinical research facility where they've tracked about 160,000 people in a population where maybe 1 in 3 or 1 in 2 individuals is living with HIV, and have extensive population level data and research data on this population. So think about Framingham size study, but this is specifically thinking about HIV and TB. And these 2 research facilities are at the heart of the twin epidemics of HIV and TB. So by comparison, if we think about the relative prevalence of HIV in 35 to 54-year-olds, you can see it's a relatively small percentage of the population in the United States, but in Kanyikuda district, where the Somkhele site for ARI is based, in that population, 1 in 2 individuals is living with HIV. So if you are interested in doing HIV research, this is truly, I think, the best place in the world to be able to collaborate and work, because we have a world-class research facility, outstanding clinicians and surgeons, and also, unfortunately, a prevalence of HIV that allows you to focus on some unique things. The ARI Tower, or the KRIT Tower, is co-localized with Caprisa, which if as an HIV provider, Caprisa has done a number of the great clinical trials in HIV, the HIV pathogenesis program, and also KwaZulu-Natal research and sequencing platform, which is where they discovered the Omicron variant of COVID-19, just as a bit of trivia. So I think it's really important to recognize we're going to start with the team in Durban and then talk about the transition to University of South Florida and the outstanding team that we have here. And I think it's really important to recognize that these efforts really rely on a large number of individuals. So while I'm very lucky to be able to work in this environment, none of this would be possible without the clinicians, the research nurses, the project managers that we work with. So I certainly want to recognize Dr. Sabia, Ms. Mtabela, Ms. Madziva, Dr. Abdul, and Dr. Kuhn as key individuals who've been involved in making the connecting us to the communities that we're able to work with in South Africa. So this is now 10 years of collaboration with these groups that has really been a very rich opportunity to learn from the communities we're working with, learn from our collaborators in South Africa and abroad, and push the science forward. So our major focus again is in KwaZulu-Natal in South Africa, and we have 5 sites with ear, nose, and throat clinical clinicians in these 5 different sites who are really helping us connect to patients and specifically to tissue samples. And we'll talk a bit about that in a bit more detail in a few slides. But tissue is a major focus of the work that we do. So I just upfront want to make sure that we recognize the large group of individuals who are assisting us. And then we have some new partnerships. And so this is my partner, Julia Toman, and I working in Malawi to develop some new opportunities to collaborate again with researchers, clinicians, and communities on studying HIV and the effect it has. So I want to take a little bit of time and just do some background on the HIV epidemic and sort of set the stage for the research that we do. And so these are 2 really, I think, tremendous and powerful photographs that tell a very brief story of where we were and where we are now. So certainly, as Dr. Lozama said, I was in school for a very long time. I think I was in 36th grade when I finally graduated and got my first position as an early investigator. But on the left, we see a photograph taken early in the HIV epidemic where there was a huge community-based protest to talk about the development of therapies for antiretrovirals. And so this is a protest being held outside of the FDA, talking about how the rollout has been incredibly slow and that there was a feeling that there wasn't being enough done to take this seriously and push forward the research. And on the right, we have Tim Brown, who was the first individual who was fully cured of HIV. And so, we've gone from a time where, unfortunately, HIV was a relative death sentence, and the time from diagnosis to an individual passing away could be as short as a year, to a time where not only do we have incredibly highly active antiretroviral therapy where we might diagnose somebody at the age of 18 and expect them to live to be 72, but we also now have instances where we have cured people fully of HIV. And so this is another photograph. So this is Ravi Gupta, who's at Cambridge, and he's a friend and a collaborator of mine, and it's a very Ravi Gupta thing to say. So this is a Times article where he says, "Yeah, I've cured a man of HIV." He was involved in the London patient's care and progress, but now I have to pick up my daughter from school. So not to suggest that cure is now blasé, but that we've really come an incredibly long way in terms of the management of HIV. There are 4 cases of HIV that have been that are considered a cure at this time. In each of these cases, you had a patient who had leukemia or lymphoma, and so not only did they have HIV, but needed treatment or qualified for treatment for these leukemias or lymphomas that occurred through a cord blood transplant or a bone marrow transplant. So the idea in each of these cases was essentially ablating the immune system and giving the patient a new immune system from a donor who had a Delta 32 mutation, which is a mutation that allows some percentage of the population to be resistant to certain strains of HIV. So essentially, we took away the patient's immune system, including the cells within the circulation in the bone marrow that would have been infected with HIV, and reconstituted an immune system that was, essentially much healthier than it may have been, and also resistant to new HIV infection. So this is not a generalizable cure strategy because the medications for HIV are now, so excellent, and the side effect profile is, so preferable that it would be, a bone marrow transplant would be I think in a much more challenging option for treatment. So let's talk a little bit about HIV reservoirs, because this is something that, is more complicated once you start digging down a little bit and trying to understand, why is HIV such a hard thing to cure? And so we often think about there being anatomical, cellular, and molecular reservoirs for HIV. In the anatomical sense, we have a CNS reservoir we're going to talk a bit about. We certainly understand the blood compartment relatively well by comparison. The GI tract, the genital tract, the bone marrow, the spleen, and even lungs and lymphatics. And we focus a lot on lymphatic system in our lab. There's also cellular reservoirs. When we talk about HIV care, most people are familiar with the idea of checking a viral load and checking a CD4 count. And this is a peripheral CD4 count in the blood that we're checking as a way of understanding how healthy a person's immune system is. And the reason that we check CD4 T cells is that HIV requires CD4 as a receptor in order to set up an infection. So the CD4 T cells in circulation are the most sort of well understood cellular reservoir for HIV in the body, because it's relatively easy to get blood and monitor and check and identify the impact of HIV and the interaction of HIV with these CD4 T cells. But there are multiple other cells that can be infected with HIV. So we're going to talk a lot about T follicular helper cells later on, but certainly astrocytes, dendritic cells, microglia, some of which are found in the central nervous system, and macrophages have all been demonstrated to be infectable, at least in the in vitro setting. And there's some debate about macrophages, for instance, in terms of whether they are a real component or part of the HIV reservoir or not. But certainly there are multiple different cell types that can be infected. And then there's a component of a molecular reservoir. So HIV is a retrovirus, and therefore it reverse transcribes its genome, but also inserts its genome into our genome. And so there are cases, we often think about viruses, making RNA, making viral proteins, and making new virions, which we see on the right side in the middle, which is a persistent infection. But HIV can also just be, the gene can be inserted into our genome, but it can be transcriptionally silent, which means it's not making virus, it's not making viral proteins. And so if you think about this from an immune surveillance standpoint, the immune system can identify cancerous cells, right? So you have CD8 T cells that are cruising around looking for cancerous cells or virally infected cells. And that's because there are changes in the cell surface that occur in both of those circumstances that the immune system can say, hey, this is not one of my cells, or it's a cell that's been infected. But if you have a latent reservoir where HIV has been inserted into the genome, it's not making any proteins. And so it's essentially invisible to the immune system and to immune system surveillance. So that's a really unique challenge about retroviral infections, is that we could clear all the cells that are actively making viruses, but one of these latent cells could later wake up, start making new virus, and set up the infection again. Another thing that we're going to talk less about today is the replication defective cells. So these are cells that have insertion of portions of the HIV genome, but not entire HIV genomes, so they're able to make some viral proteins, but are not able to make a full virus. So that's changes a few things on the research standpoint, and a little bit in the clinical care standpoint, but we're not going to focus as much about it today. I'm going to talk about the CNS reservoir just a bit. This is not a major focus of our lab, but it's a really important idea to understand HIV and the goals of HIV cure. And the CNS reservoir is unique because of the blood-brain barrier. And so HIV can transition into the CNS in a couple of different ways. So there's a free virus transfer across the blood-brain barrier, which can occur, but also cells, monocytes, and T cells that are able to cross the blood-brain barrier can also carry virus across as a Trojan horse. And there's multiple different cell types within the CNS that can become infected with HIV. This is important because there's been demonstration that you can develop different populations of virus in the blood and in the CNS. And so in this case, starting with A, we have HIV that transitions into the CNS by one of those two mechanisms. You then start a patient on antiretroviral therapy in B, and you decrease the peripheral the peripheral population of virus that's circulating. But in some cases, the antiretrovirals don't cross the blood-brain barrier quite as well as we would hope, and so you may have incomplete suppression in the population on the right-hand side. And over time, you may develop a mutation, and you develop a mixed population that we see in C, which is within this CNS reservoir. And you may have then back transition of a mutated version of the virus back across into the periphery that we then would detect as a mutant that is resistant to the antiretroviral therapy that the patient is on. And this has been demonstrated. So this is some really fantastic work by Ron Swanstrom's group looking at distinct differences. And so here they're looking at the clones that are identified within the periphery and within blood and within CNS component checked by CSF. And without sort of delving too deeply into the figure on the right, what you can see is a clonal population that's distinct and different in blue and sort of the 11 o'clock position of that of the graph of the figure on the right, whereas there are multiple other sort of mixed populations externally. So there's definitely distinct differences in the components that can be seen in certain patients in the CNS and blood. So this is a real consideration, and it becomes important to think about from a research standpoint and a clinical care standpoint, what does that mean, both for treatment and for cure? We have a collaboration through one of the Martin Delaney collaboratories that I belong to, the Pediatric Adolescent Virus Elimination Network, with FAMCRU, which is the Family Center for Research with Ubuntu and University of Stellenbosch in South Africa, who also do a lot of work on CNS reservoirs. And so definitely want to discuss Mark Cotton and his team quickly at FAMCRU, who've really been looking at latent reservoir characterization and neurocognitive function in some very exciting ways. So again, just want to talk a little bit about viral genome integration and fate. So if you have HIV provirus, which is after the genome for HIV has been inserted into our genome, we call that the provirus. So it's not a virus yet, but has the potential to become one. You can have transcription of active HIV virions, which we see in the bottom. You can again have that latent reservoir where it's transcriptionally silent, and you can have defective viral proteins that are being made. So looking at this as sort of same idea in a slightly different way. In the very middle here in green, we have a CD4 cell that's activated that gets infected with HIV, reverse transcribes and integrates its genome, and then you have transcription, which when the cell is making virus, it can be it is visible to the immune system, and so you can have immune clearance and cell death. But notably, these cells can also become quiescent and become latently infected, so their transcription shuts off. Whereas at the top of this figure, we see a direct infection of a cell that's in G0 sleep memory state and a latent reservoir cell in the top, and this can also happen in TRM cells as well. So, when we think about clearance and cure, we are able to target cells that are transcriptionally active, but it's really challenging to think about targeting the latent cell population. And so, there have been some advances in using CRISPR-Cas9 to maybe try and snip this reservoir out, but it remains a major challenge in terms of the cure agenda. I want to just also mention that measuring the reservoir in HIV is relatively complicated. HIV is an RNA virus, and so it uses an RNA reverse transcriptase, which tends to make a lot of mistakes, compared to a DNA reverse transcriptase. And so you have, in many cases, incomplete genomes or genomes with errors that aren't able to make fully functional HIV viruses. And so if we think about the various different ways to quantify the amount of virus that's in a human body, measuring the reservoir becomes relatively difficult. On the more stringent sort of classical side, we can use a viral outgrowth assay, which is where we essentially stimulate and try to make viruses, and we measure how much virus can you make that can then infect another cell. And this only measures the reservoir of complete virus, but may undermeasure because not every cell that's potentially capable of making a virus is going to be measured within the viral outgrowth assays. But if we go to a more inclusive measurement of the reservoir using PCR, these little bits and pieces of HIV that are integrated will get picked up, but they aren't necessarily capable of making a full infectious virion. So there are difficulties even in just trying to say how big is the reservoir, and we over and underestimate the functional reservoir pretty consistently. So let's delve into cure strategies, and then we'll switch over to the work that we do directly in my lab. But there's a number of different ways that we've been thinking about trying to cure HIV. So if we start at the 7, 8 o'clock position, immune checkpoint inhibitors have been evaluated and are being evaluated in our lab, but using things that change the function of CD8 T cells and natural killer cells like anti-PD-1, anti-CTLA-4, IL-10 to increase or change the way that the body's immune system responds to viral infected cells has been a strategy. Using therapeutic vaccines, which induce and again sort of augment CD8 T cell responses, moving around to about the 11 o'clock position, thinking about augmenting natural killer cells, dendritic cells, macrophages, neutrophils to recognize cells that are infected with HIV. CAR T is a new approach that's being used. I think is really exciting to take T cells and give them the ability to specifically recognize or track into areas, into some of these reservoirs in unique ways. Interferon alpha is an approach that we're starting to explore with Bob Gallo and his group here at USF, and thinking through the effects of interferon alpha as an antiviral mechanism, sort of a general antiviral mechanism. And then we're going to talk a little bit more about latency reversal agents in the next slide on the next position. So there's two basic approaches. If you have a latent cell, so again, this is a cell that's not making HIV, but has the ability to do so. One strategy is what's called shock and kill. And so the idea is that you can stimulate and wake these cells up and use a latency reversal agent that then takes a latent cell, makes it an active cell. So now it's making virus and it's expressing viral proteins on the cell surface, and now it can be targeted for kill. And so that's the sort of classic approach that's been taken over the last 10, 15 years in terms of clearing this latent reservoir. But there's a newer approach, which is block and lock, which a number of different collaborators and of ours at PAVE have been invested in, where the idea here is to use proteins that instead of taking a latent cell and waking it up or using CRISPR-Cas9 to cut the genome out, essentially you just take that latent cell and put it permanently to sleep. So the idea is that using some proteins we'll talk about on the next page, you block the ability of HIV to wake up, and then you lock it into a permanently latent cell. And so this is not a cure necessarily in the same way that completely eliminating all HIV genome from the body would be. It's a functional cure where if we put every latent cell completely to sleep and then clear the active reservoir, and the patient never needs antiretrovirals again, never develops a viral load, that's a really good outcome. And so it's a new approach that's being taken. So we'll talk quickly about what the block and lock strategy is. Again, I recognize this is a lot of basic science in some cases, but trying to just pull the major message out here. In general, there's a protein complex called LegF P75, which interacts with the HIV pre-integration complex. So what HIV pre-integration does is it finds areas within the genome to help insert that HIV genome into an area that's relatively transcriptionally active. So the HIV virus, at the expense of anthropomorphizing, wants to be in an area where it's going to eventually be transcribed to some degree. And so the LegF complex helps target it to areas usually around histones that are going to be sort of folding and unfolding and eventually be transcribed at some point. Using legins, which is a great name for a protein, the legend proteins interact with LegF and essentially target the pre-integration complex to areas that are incredibly transcriptionally silent. So what you're doing is you're burying HIV in areas of deep latency that ultimately, we block and then lock the complex in such a way that it doesn't become transcriptionally active again. So all this to say, a lot of really interesting strategies in terms of clearing the latent reservoir. I'll just quickly also talk about broadly neutralizing antibodies as another cure component. When you're infected with a flu strain or a COVID-19 strain, you make antibodies to the strain that you have. In some cases, in HIV, what's really notable, of course, is that there's a lot of mutation, and you have clones of HIV that develop even within an individual that have a relative breadth of sort of different antigen display. And so when you're infected with HIV, you tend to make antibodies to the HIV, but it already mutates. So by the time you have functional antibodies that target that initial clone, it's already escaped and moved on to make new clones. And so what broadly neutralizing antibodies are they are antibodies that target and can neutralize a lot of different kinds of virus and a lot of different viral clones. And these aren't made by everybody in high amounts, and finding individuals who make really potent broadly neutralizing antibodies is another strategy for cure. And so here we're just sort of showing conceptually how that might work. So this is the GP120, GP41 interface. So this is on the outside of the HIV virus. This is the part of the virus and the protein of the virus that interacts with cells and is essentially the key that allows HIV to enter cells. And so what you'll see is it's a relatively complex structure. And I think about this a lot of times as a tree. So you have, sort of a trunk, and then you have, major branches, but then you have foliage and leaves. And HIV is able to change the leaves relatively easy. And so broadly neutralizing antibodies, conceptually, I often think of as targeting areas that maybe are less able to change. So you might target a major branch or the trunk of the tree, or an area of foliage that's just unable to change very easily. And so by using a combination of broadly neutralizing antibodies, we may be able to target HIV clones that are incredibly diverse, so essentially multiple different variations of this same tree structure. And so, that's another major approach that's being taken. So as we think about HIV cure, it's a relatively complicated thing. If we think about the reservoir for HIV in general, there's tissue, cell, proviral components to this as well. And so the overall cure strategy is nuanced and requires a lot of research and collaboration between a bunch of different groups. So I think with that, I want to move on to the work that we're doing as a part of this overall cure strategy. And we are really interested in tissue reservoirs. So not just necessarily CNS reservoirs, but the lymphoid tissue reservoir, which we're going to continue to talk on in a bit more detail. So historically, most work in HIV, most research has been done in blood because blood's relatively easy to acquire. If I can ask a participant who's living with HIV for blood, and I might give him, travel voucher and maybe some money for time and convenience, but blood is a relatively easy thing to obtain. But if I ask somebody for a brain biopsy or a lumbar puncture or, can I have a lymph node, that's a much more challenging thing. And yet, I think we now know that we've been studying HIV in blood for about 40 some odd years, and there are reservoirs that require us to understand the tissue. And so we really, in our lab, function a little bit in human clinical trials, but work in these explant and organoid cultures that I'm going to talk about in more detail, and some 3D tissue cultures. And we have some collaborators who work in animal models to try and understand how virus interacts in tissue in ways that's different from blood. This is a complex figure, but I'm going to go through it because I think it sets up the rest of the talk. And I'm just going to highlight the parts that I think are most important. We work largely in tonsils because tonsils are lymph nodes. They have a few funny aspects to them. They have a mucosal surface that's, that maintains colonization with bacteria, and yet they are lymph nodes and have classic lymph node structures. And the basic job of a lymph node, and what we see down here in the bottom left-hand corner, is a piece of tonsil tissue. We've stained this by immunohistochemistry with an antibody to MCM2. Ultimately, all that does is any cell that's dividing is going to upregulate MCM2, and in this figure is going to be brown. And so what you see in the tonsil tissue is a lot of these little thumbprints, and those are germinal centers. Now, germinal centers within lymph nodes are responsible for antibody production. And they do this through a process called somatic hypermutation and affinity maturation, which is pictured on the right-hand side. So ultimately, there's a factory, which is the germinal center, and its job is to produce a plasma cell, which we see at about the 2 o'clock position. Now, a plasma cell is a B cell that makes antibodies. So, the goal if you were to get a flu vaccine, is that you then have plasma cells that recognize the antigen that was presented to them and are able to make a ton of IgG. We also produce memory cells. Now, the way that works is through a cyclical process that we see in the center, represented in the germinal center. So essentially, starting at the bottom, you have 3 key players. The first is a B cell. And the B cell's job is to make antibody. And so you have a B cell that then goes up to this FDC, which is a follicular dendritic cell. FDCs are professional antigen presenting cells. So when you get you get a flu vaccine, it takes a piece of that antigen, presents it to a B cell. And if the B cell has a strong enough affinity for that antigen, it gets support by this little purple cell, which is called a T follicular helper cell. And essentially, if the interaction is strong enough, the TFH cell gives it permission to go through another cycle. The B cell then goes back down to the bottom. It proliferates, but also mutates slightly. And through multiple processes, you get a selection that's called affinity maturation. So essentially, you have B cells that are getting better and better and better at recognizing antigen, and then eventually become plasma cells or memory cells. So all that to say that TFH cells are really important in terms of selection, FDCs are really important, and B cells are really important. I've often thought about this as sort of a Roman Colosseum where, you have the FDC is presenting the antigen and this is the lion or something, and the T follicular helper cell is sort of your emperor, which is giving a thumbs up or thumbs down, and the B cells are the gladiators. But, whatever way that sort of helps understand the process, essentially it's a selection for stronger and stronger affinity. Now, this is important because germinal centers are really unique as an HIV reservoir. So this is some work done by Manuel Asawada, who is a postdoctoral fellow at RE, working in collaboration with Henrik Cloverpers, who's a close collaborator in our group. What we have on the bottom is a person living with HIV. On the top is a person living without HIV. This is immunofluorescence. And if we start with the right-hand column, this is P24, which is a component of HIV. So essentially, things that glow green. Things that glow green. Things that glow green. I have a little bit of feedback there. You guys getting that? Let's see. So anyway, P24, what we see in the top, there should be none because the person is not infected with HIV. On the bottom is a person living with HIV, and we see that most of that P24 is within the germinal center, that little thumbprint. And it's important because if you look at the left-hand side, this shows CD8 T cells. Now, CD8 T cells are the cells that help to kill virally infected cells, and what you see is they're largely excluded from that germinal center. So what we have is essentially a sanctuary site where the cells that would normally clear out the infected cells are largely excluded. And this is probably important just to keep CD8 T cell signaling and natural killer cell signaling from changing that component within the germinal center. But it also provides what we think is a really profound and important anatomical or subanatomical reservoir that we're focused on in our lab. This is some additional imaging that I'm gonna pass on, but just showing in somebody living without HIV, somebody living with HIV in antiretroviral controlled situations, and then viremic patients. There are some nuances and differences, but certainly we see that CD8 T cells are present more outside of a germinal center than inside of a germinal center. Now, to start to understand this and look at tissue, we do need sort of the variation of patients living without HIV, patients living with HIV who are on therapy, those who are off therapy, and sort of every sort of different variation that we might be able to think of, including comorbidities and age, to try and understand this in much more detail. So over the last 10 years, Henrik Cloverpers and Al Leslie, who are two researchers at the Africa Health Research Institute, and our lab have been working on creating a tonsil cohort in South Africa. And so this, our youngest patient, I think we have tissue and blood from a patient who was 1 month old, somebody as old as 56. I think we might even have a 62-year-old individual. So across the age spectrum, and we have both controls and people living with HIV. But what's really notable is this is the largest tissue biobank of HIV in lymph nodes or in lymphoid tissue in the world. And the reason that we can do this is that we unfortunately have a large burden of HIV within the populations that we serve. So by comparison, if we think about the, say, 2,500 people who are living with HIV that we take care of at USF and TGH and Department of Health, if we were waiting for them to get a tonsillectomy, how often is that going to happen? Out of 2,500 people in a year, maybe we have a couple of tonsillectomies. In South Africa, if you're in a population where 1 in 4, 1 in 3 people may be living with HIV, the opportunity to obtain this tissue is much greater. And in this case, these are all medically indicated tonsillectomies, so we're not doing this for research purposes. We're essentially just asking if we can have the tissue after the tonsillectomy is completed. And also notably is that most of these tonsils are coming out for snoring. So this is for sleep disordered breathing. So these are not infected tonsils. These are usually just large tonsils that may be contributing to the sleep disordered breathing. Our cohort, and this is just a subset that I'm currently working on at the moment, includes both male and female patients across a various diverse age spectrum. We also have a diverse viral load spectrum from completely controlled to moderate viral loads to viral loads that are quite uncontrolled, as well as CD4 counts from 0 up to normal levels. So now let's dig into some of the work that we've been doing, and we're going to talk a bit about germinal centers and germinal center health. So again, these are both tonsils on the left and right-hand side. This is H&E staining, which is a very basic sort of stain to show cells and differences. In the sort of purple areas, that's the tonsil follicular tissue. And what you see on the left-hand side is a lot, again, of those little tiny thumbprints. And on the left is a 41-year-old female living with HIV, CD4 count of 305. But most of those germinal centers look about the same in terms of size and shape. Versus on the right, we have a 17-year-old CD4 count 512. And we see that there's a variation in the sort of size and shape of germinal centers. So some very large ones, some that are shaped like barbells. And this is a phenomenon that was originally described in the late 1980s, early 1990s in autopsy cohorts, but really hasn't been done since that time. But we know that initially with HIV infection, you have hyperplasia or enlarging of germinal centers, and then they sort of get funny shaped and then they disappear. So we also have patients where there are no germinal centers at all. As we think about the ability of a person living with HIV to respond to an infection, certainly the peripheral CD4 count is really important, but I think these changes are really important as well. And if you think about germinal centers as factories for production of plasma cells or production of cells that can make antibody, changes in the sort of factory matter. So how big is the factory? And then you think about, well, how many workers are in the factory and how active are they? So the changes, I think, are really important. So we've developed a really complex approach to analyzing the amount of available tissue and the size and shape of germinal centers and their cellular components. This happens if you have really good medical students and volunteers and staff in the lab who are willing to go through and process these images. And these are really large images with hundreds and hundreds of them. A lot of this is time spent circling germinal centers and then looking and trying and optimizing various different stainings. So John Peyton Bush is a fourth-year medical student who worked with the University of Kentucky and continues to work with me remotely. Derek Jacobs is an MD PhD student who's joined my lab full-time, who's done a lot of the early work on H&E evaluation. But we definitely see that there are some differences in both the size and the relative compactness of germinal centers in persons living with HIV and not. And then we want to go through and understand the potential reservoirs within cells. So we have, in this case, a 40-year-old male who's living without HIV, but this was a nice example of some immunohistochemistry. And so we essentially have multiple slides that we've taken, but they're sequential slides. So this is the same germinal center in each case. Overall, the germinal centers look healthy. They're low variability in size and shape, but we can start to look at the T follicular helper cells, which is sort of that emperor who decides if a B cell gets to go on or not, which stains with PD-1. Macrophages are a different potential reservoir, staining with CD68, and the follicular dendritic cells, which are those octopus-like shapes that are presenting antigen. So we can now start to sort of quantify not only the size and shape of the germinal centers, but their content. And this can be done with some automated programs that detect staining. So you have to have somebody who's good at cutting and staining. And so Derek and some other members in the lab, Lily Polans, have become gained quite a bit of expertise in cutting and staining and optimizing these things. But we can now automatically count the number of PD-1 cells and CD68 cells. And we see that there's definitely differences in terms of people living without HIV, which is shown in black here, and those living with HIV in terms of the percent of CD4, the percent of TH cells within a germinal center, or macrophages and various other different things. And notably, this changes based on the age and the CD4 count of individuals. So we know actually that the germinal centers do change with age. And so as individuals get older, the number of PD-1 positive or T follicular helper cells, those emperor cells, does decrease. And we are seeing now for the first time that also the number of cells decrease as the CD4 count goes down. So that's in the other direction with the CD4 count that is lower on the left-hand side. But this raises some really interesting questions about, what is the impact of HIV on the ability of the body to make an antibody. So as a individual who cares for persons living with HIV, if I give a vaccine to somebody who's living with HIV, I often think about their response to the vaccine may not be quite as robust as it would be in somebody who's not living with HIV, and that's independent of their CD4 count. So they can have a really robust CD4 count, and there's still times that they don't respond quite as well to a vaccine or have the durability of vaccine protection as I might typically expect. And so, that may be worsened with increase in CD4 count, but it's interesting that we also see a very similar thing. So if I think about vaccinating somebody who's in their 70s or 80s, they seem to also have maybe less of a robust response to vaccination. And I think that there's probably an overlap in terms of mechanism, that we see some of the same changes in our germinal centers, which are factories for plasma cells, in these T follicular helper cells and also in follicular dendritic cells. So those two major components that are helping select good B cells. So I think we may understand something about immune senescence and just the changes that happen in the immune system with age by studying the impact of HIV on these cells, because we can now control through infection or vary the number of these cells in persons living with HIV. And I think that's an R01 that we just recently submitted out of my lab, is exploring the underlying mechanism between these two phenomenon. I'm going to skip through this slide, I think, and just show that this is something that we've been actively engaged in, is trying to really understand this reservoir through immunohistochemistry. And ultimately, we're seeing that the P24 signature that we see here in the top left-hand side seems to have, in this case, the brown staining does seem to be largely between the blue nuclei, right? So this seems to be between cells. So whereas a TFH cell has a staining pattern that looks, relatively robust like a T cell, and a macrophage is slightly larger and maybe a little bit extended in certain directions, the follicular dendritic cell staining pattern looks like this sort of octopus again that's intercalated between all the different cells. And so I think this highlights the fact that a large part of the reservoir may be associated with follicular dendritic cells, and teasing apart how that changes over the course of the HIV epidemic is a really important first step in characterizing this lymphoid reservoir and trying to understand how to maybe mitigate it. What's notable about this is even though these are not maybe the most complex experiments that you could imagine to do, they're incredibly unique, because up until this point, having access to this tissue has been incredibly difficult, right? So it requires this overlap of having a community that you can engage with that is living with HIV, but where there's a large burden of disease, so that you're able to identify these rare patients more than just a couple times a year, but obtain enough sample to try and look at the effects of age and viral load and CD4 count as a major component of these changes. And so it's really critical to have had these very long-standing partnerships in places like South Africa and Malawi that really allow us to bring together a team that can make this kind of evaluation possible. So, I fly to South Africa relatively routinely for short trips, but it's because there are these opportunities to partner with these communities in ways that allow us to ask some very fundamental questions that I think are critical. Just want to look at sort of some challenges in terms of, looking at the reservoir. So these CD21 follicular dendritic cells are not individual punctate cells that are easy to pick out, but are less discrete and sort of look like giant octopi. So, how do you count the number of cells in this circumstance? So some other things that we've optimized in terms of being able to compare one patient to the other. I'm going to move on to a couple other things that we do. So, I think it's critically important that we understand the underlying just architecture of a lymph node or of a tonsil cell in order to understand the reservoir. So you have to sort of start, big picture and say, what is the landscape for where the HIV reservoir might exist within individual patients? But then we really want to move into functional studies as well. And so we use a lymphoid organ explant and also an organoid model where we start with a freshly removed tonsil here on the left, and then you can embed that living tonsil tissue into agarose, which is low melt agarose, so it never really heats up all that much. And you can cut it in a very thin slice. And so this is a living piece of tissue that was, we usually have it from the operating room to in the hood in the lab within an hour. So this is a living piece of tissue that you can then culture in a dish. And what's amazing is that this, in a dish, it functions as a lymph node. You have to cut it very thin so that it remains alive in the system. But this is, now experimentally modifiable, but it's a completely physiologically relevant system because we haven't changed the architecture. All the cells are in the same place in the right spot, including the germinal centers. And what's really remarkable is you can take a flu vaccine, drop a microliter or two into this cell, this system, and the tissue will make an antibody in response. And so that's seen here. They take a piece of tonsil tissue, embed it, thinly slice it, and you have an explant culture. Alternatively, that has to be done day of, so right at the time of surgery. But you can also take and process tonsil tissue into single cells, mononuclear cells. You can then freeze those, and so later you can come back and do a comparison with multiple different individuals. But if you plate out these mononuclear cells and give them a little bit of antigen, the drive to make antibodies is so strong that they form germinal centers within a dish and will also make antibody. So we see in this case, taking a tonsil out, putting it into a culture system, giving it an antigen or an infection, it will make these germinal center structures. And here we see it's able to produce IgG and IgM, that affinity matures over the course of 3 to 4 weeks within the system, and you're able to produce, in this case, this is to influenza vaccine. But you now have a system that you can see not only how is the infrastructure different within the germinal centers, but how functional are they? So if you have an older individual, do you make less antibody? If we have more HIV reservoir, less HIV reservoir, and we can acutely infect this with virus, so we make virus in the lab. We can look at tissue from persons living with HIV, and we can start to control based on age and sex. To couple with this, we use a lot of spatial transcriptomics and live tissue imaging approaches that are pretty fascinating, that I'd be happy to talk in more detail about, but really delve into the basic science quite a bit more. And we're interested in, again, sort of this impact of viral infection. So we make a virus, we infect a piece of tissue from a control patient that we might recruit from TGH, use some antigen to stimulate to make an antibody, and then we have a suite of different approaches that we use to try to characterize the ability of that tissue to respond to antigen. I want to switch gears just a little bit for the last couple minutes of the talk and say, a lot of this really relies on our ability to engage with patients. So as a physician scientist, I think, trying to think bench to bedside and back as much as possible is really critical. We have a number of different sites where we're obtaining samples. So most of our samples from persons living with HIV come from KwaZulu-Natal, but we have a new cohort that we're launching in the Western Cape of South Africa and also exploring in Lilongwe, Malawi. And we have a biobank now that we've launched here at USF that we're using to power this work and also work of a lot of our collaborators. And I want to talk just a bit about cohort acquisition. So we're now switching gears to think about how do you recruit patients for this kind of opportunity. And we're going to talk just about patient recruitment in general, for instance. So on the left-hand side of this figure, we have various different ways as an infectious disease provider, I might see a patient. So whether this is inpatient, an outpatient, from a referral from a non-infectious disease provider, somebody in the emergency room, somebody who self-refers, somebody who sees an ad and refers, or word of mouth might be ways that patients who are interested in participating in research might sort of present themselves. On the right-hand side, we have multiple different kinds of infectious disease research projects. And typically what happens is that every time you want to do a research project, you start from scratch and you have this sort of ad hoc recruitment, which works and is the way that it's typically done, but is maybe less efficient and sort of redundant in certain circumstances. So one of the things that I'm really involved in is centralizing recruitment efforts for research. And so within the division, I've created what's called the FIDER registry, which will, in collaboration with Charlotte Sambunwit and Kami Kim and a number of other individuals, which is a way to take all of these different patient streams, place people into a registry that says, I'm interested in any infectious disease research, I'm interested in HIV-specific research, et cetera, and then using that as a way to then seamlessly enroll into multiple other projects. This is based on some work I did at the University of Kentucky. So I founded the Kentucky Infection Treatment Excellence Registry, the KITE registry. I think you need a good acronym for people to remember. So we used KITE. And essentially, the FIDER registry works very similar to KITE. This was in the case of the KITE registry, people would be referred or would self-refer or use a QR code or a link. Takes them to a REDCap information gathering survey, about 5 minutes long, gets demographic data. In this case, it was a COVID-19 registry, so vaccination status and history, et cetera. We were this is passive. What's amazing is that most of the time this happens very passively. And so just month by month, we recruited patients into the KITE registry and have just launched the FIDER registry and are doing the same thing. But this correlates with some translational research infrastructure that I built at Kentucky and I'm building here at the University of South Florida, that the combination of the registry and just a very basic translational research infrastructure, we were able to get a $4 million long COVID grant. And then that has led into 3 additional R01 equivalent grants just by having this sort of seamless infrastructure. Because instead of saying, I'm sure that we can recruit 200 people, I could say, I have a list of 2,000 people who are interested in participating in COVID research tomorrow or HIV research tomorrow. And so we were highlighted as in the national Recover initiative for long COVID as a site that was very successful in acute enrollment because of the registry. So I want to just quickly end with the fact that we've launched the FIDER registry, which is the Florida Infectious Disease and Global Health Team for Emerging Disease Response, that allows us to, again, just refer individuals passively, or we can have providers in internal medicine, emergency department, the ID department, the ICU can refer patients into the registry. It's a very short form that is either filled out by the provider or by the patient. And then this powers the ability to do a tremendous amount of different research. And this is very valuable for my research program, but is intended as a generalizable resource. So if we have a fellow in ID who wants to do a project, they don't have to start from scratch. Or if we have a junior faculty or somebody in internal medicine who's interested in this case, it's an infectious disease registry, but interested in Staph aureus bacteremias or, essentially anything, this infrastructure is now IRB approved at USF and TGH, and we're moving into the Department of Health, but is approved and is, we just need to populate it with individuals, but then becomes a really powerful platform for writing grants and conducting studies. I'll recognize Danielle Curtis, who's a project manager in our group, for all of her work in helping set this up. But we have a lot of information out there. There's QR codes. You're welcome to scan it now. This can, anybody can join the fighter registry. It doesn't matter. We can hang things on bus stop, benches, radio platforms. We can talk about it in forums like this, but anybody can sign up for REDCap and then can be recruited into studies, whether it's a biobanking study, survey study, observational study. So I wanted to give a quick plug for that. With that, I want to say we have a tremendous number of collaborators and acknowledgements all over the world. I want to specifically recognize the individuals in my lab here. So Danielle Curtis, Derek Jacobs, Lily Polans, who are project manager, MD PhD student and PhD student in the lab, Hanani Ibrahim, Janavi Sabarwal, Brandon Lopez, who are undergraduate research volunteers in the lab, and John Peyton Bush, who continues to work with us. Have a lot of funders. Specifically want to highlight the Pediatric Adolescent Virus Elimination Collaboratory, but a number of different funding support over the course of the years. And with that, just say thank you, and please do feel free to reach out if there's any additional questions. So with that, I will stop sharing and take any questions that you might have.
Feel free to take the mic if you have a question.
Zach, would you mind share our colleagues how they can refer their patients to the fighter registry?
Absolutely. So what we're just getting started in terms of, working with various different providers. But what we hope to do in the very near future is send out sort of a larger email to the provider network with some of the information. We're going to start hanging some of the flyers in key areas where we might encounter patients who might be interested or where providers might be able to see it. We have links and QR codes and advertisements. So my hope is to begin to work sort of with individual groups within our provider network on a smaller basis to get this launched. But I'll certainly hoping to send out a much larger email about it in the very near future.
Dr. Kim?
So thanks, Zach, for the great talk. I just wanted to sort of dovetail on, some of Zach's remarks about what we're trying to do. Obviously, we're very lucky in that we're very involved within the community as far as HIV care, and we're working very hard to enhance our clinical and now basic research in HIV. But we're also using this as a mechanism, given Charad and Zach's expertise, to build infrastructure, across the group. So Les Shaw, John Adams, and I have a pilot project to look at AMR research, so antimicrobial resistance. So we are working, and a number of our faculty are already involved with that, Suzanne Silbert, Les Shaw. And so we are actually taking advantage of the infrastructure that Zach's setting up as far as the fighter registry, to develop the infrastructure for all sorts of different projects beyond his HIV study. And this is also something that funding agencies love to have a registry and a biorepository, but they frequently don't want to pay for it. So this is where, if you have an experience in global health, you know how to do things efficiently and on the cheap. So, obviously, so we're going to extend that to other things that are very mission critical, like hospital-acquired infections, HIV, and other, aspects of clinical research. But for other people who are interested in other important translational projects, we're happy to work with all of you to sort of figure out how to get students and trainees involved in these kinds of projects and develop an infrastructure so that, you know, the entire collective group of us who have a lot of interesting questions but are often too busy with clinical service and things like that can really start mentoring all of our very excited and motivated learners into actually starting these kind of work. So, so this is, obviously, Zach's done a tremendous amount in the short time he's been here, but this is also part of a greater group effort within our division, both at USF and at TGH. And we really welcome the opportunity to scale up these opportunities in other divisions and other units across the university.
Thank you, Dr. Kim. And I agree. I mean, the registry is intended as a generalizable resource. So it's something that, you know, we really want to make available to the wider USF research and clinical community. So. All right. Well, at the top of the hour, you know, please feel free to reach out individually if there's any questions that you have or any additional discussion points. But I really want to thank everyone again for their time and attention. And again, Ms. Newcomb, thank you for your assistance in helping put this together. And yeah, wonderful. Thank you all.
Thank you, everyone.
Thank you. Great talk, Dr. Porterfield.
Great talk.
Thank you.
Good job.